Hello and welcome to the Global Immunotalks. My name is Matteo Iannacone, and on behalf of the organizing committee, it is my great pleasure to introduce Judith Agudo as today's Global Immuno speaker. Judith was born in Sabadell, a town near Barcelona in Spain, and she obtained her PhD at the Autonomous University of Barcelona, where she studied factors involved in cell growth, apoptosis, and inflammation in the diabetic pancreas. And after her PhD, she obtained a Fulbright scholarship to perform her postdoctoral training in the Immunology Institute at the Icahn School of Medicine at Mount Sinai in New York, under the supervision of Brian Brown. And there, she first studied molecular regulators of innate immune responses and dendritic cells development and function. And then she developed a very interesting novel mouse model to study how T cells interact with any cell in vivo. And you cannot not love the name she gave to that mouse strain, the Jedi Mouse. In particular, she exploited this technology to study immune surveillance of tissue-resident stem cells in their niche in vivo. And she identified a correlation between cell cycle arrest and immune evasion. She was then recruited as an assistant professor to the Department of Cancer Immunology and Virology at the Dana-Farber Cancer Institute and the Department of Immunology at Harvard Medical School. And in her lab, she discovered a population of quiescent cancer cells in breast cancer that escape T-cell immunity and possess higher tumorigenic potential. Immune evasion is achieved by an orchestration of an HIF-1 alpha dependent immune suppressive niche. And currently, she's investigating mechanism of immune evasion of cancer stem cells to uncover how these cancer cells with higher tumorigenic potential evade cellular immunity and flourish to grow new tumors. And she also continues to study immune surveillance of tissue stem cells to identify strategies to allow survival of stem cells-derived cells, for instance, for transplantation. And in recognition of her work, Judith received multiple awards, such as the Susan Komen Career Catalyst Award, the Mary Kay Foundation Award, the Smith Family Foundation Award for Excellence in Biomedical Research, the MHC-I Junior Faculty, and the American Diabetes Association Pathway Accelerator Award. Judith, thanks so much for having accepted our invitation, and we very much look forward to your talk entitled Immune Evasion of Stem Cells and Cancer Stem Cells. Now, before I hand it over to you, it is a tradition of the Global Immunotalk to ask a question to a speaker to get to know them a little bit better, or to perhaps inspire the next generation of scientists. So the question we have chosen for you is, as an immigrant yourself, what advice would you give to a young scientist who's training abroad?
Yeah, so immigrants, so scientists are very lucky that we get to, live in other countries and travel a lot. And the thing is, after, the initial excitement, there's a moment that I think it happens to all of us, that you miss your family, you miss your country, you miss your people. And science also has moments that, things don't work, and it can be very tough. And when you are abroad and you are very far away from the people you love, it can be really, really, really hard. So my advice is that try to get a network of support as soon as you can, because at least in my case, I think I wouldn't have made it without the friends I met along the way, and of course with my family as well.
Here, yes. Thank you, Judith. I think you can go ahead now and share your presentation.
Yeah. No, is it okay?
Yes, works beautifully.
Okay, perfect. Yes, I mean, of course, I need to start by saying thank you, because it's truly an honor to be here. Like a place where so many famous scientists have presented, and now I'm here as well. It's really amazing. So thank you so much for this unmatched opportunity. And I also want to thank you for this effort and organizing these talks, which I think is a great opportunity to democratize science, because now anyone around the world can see these talks and learn about cutting edge science and immunology. And without further ado, I'm gonna start my presentation. And as Matteo explained during his introduction, I actually did not start my scientific career as an immunologist. That's a confession I have to make. Instead, I was interested back then, a long time ago, in regenerative medicine and how, to regrow tissues. And the disease model I was studying was diabetes. So my project was regrowing beta cells for treatment of type 1 diabetes. And when you work in regeneration for an autoimmune disease, you very quickly realize that even if you are super successful and you do manage to regrow whatever you want to regrow, it's actually, unfortunately, doomed to fail because the immune system is gonna go back and kill everything that you've done. So after, that sort of tough lesson, I decided that I needed to learn immunology. And what I did is I obtained this Fulbright scholarship, and I went from Barcelona to New York City to develop tolerogenic vaccines. And this was done in the lab of Dr. Brian Brown. And my goal back then was, as I said, to develop tolerogenic vaccines targeting dendritic cells. So I was very fortunate to be also co-supervised by Dr. Miriam Merad. And what happened was that actually inducing tolerance is easier said than done. So I had lots of struggles, but science, there's always an avenue, and science it's unpredictable sometimes. So my tolerogenic vaccines, didn't go where I wanted them to go, but I did do what I thought was really cool. I mean, of course, really cool science and cool findings in basic function of dendritic cells. And we published the work. And after this was done, as I said, tolerogenic vaccines and inducing tolerance is not trivial. It's not an easy task. What I thought is, okay, I'm not gonna give up, I left my country, I came here with this vision. Maybe the answer is, rather than engineering or manipulating the immune system, which is sort of like an unstable entity, you may succeed and induce tolerance, but then the immune system, there's an infection, there's something happens, you lose that effect. So why not protecting the cells in a sort of a cell autonomous manner, can we engineer beta cells, for example, so they don't get killed by the immune system? How do we approach these? Where do we get good targets? How do we learn about this? And something that I read many times when I was working in the regenerative medicine field was that stem cells are immune privileged. There's this big dogma that, stem cells are immune privileged. And you read this in a lot of reviews. And this is now, I would like to stop for 1 second to just, emphasize and tell the trainees in the audience that when you read something in a review, don't take it as, as absolute truth. Be critical, because what happened is that I went back to this literature, and one review, when this sentence was there, cited another review, that cited another review, that cited another review. And like this many times, till you ended up with a few papers showing that embryonic stem cells, for example, have low MHC class 1, and therefore they are supposedly immune privileged. Which means that basically there were no data out there, that it's not true that this big statement, had been demonstrated. And no one had ever even attempted to study whether the tissues that we have in, the stem cells that we have in our tissues, whether they are indeed immune privileged. So sure enough, with Brian, we decided, okay, this is an interesting question. Let's try to tackle it. But how do you go about it, how do you study immune surveillance or immune privilege of stem cells? And a tool that immunologists often use are TCR, T-cell receptor transgenic mice or T cells that recognize a given antigen. So for example, if you want to study immune responses against insulin-producing beta cells, you could potentially generate a mouse whose T cells recognize insulin. So it's a T cell specific model. So if we wanted to do that, and we wanted to study immune responses against stem cells, what we could do potentially is to generate TCR transgenic mice that recognize antigens that are specific for tissue stem cells. But that was very complicated. And it meant that we need to identify these antigens and probably make a T-cell mouse for every single stem cell I was interested in. So obviously it wasn't going to happen. Conversely, another approach was to take advantage of a mouse that already existed, such as the OT1, famous OT1 mouse that recognizes ovalbumin, a model antigen. And we could have this ovalbumin expressed in tissue stem cells. So we needed to make knock-in mice to express this ovalbumin. That wasn't going to happen either. And when we were about to give up, we actually realized that stem cell biologists, they love GFP. They love the green fluorescent protein because it's a great reporter. And there are lots of, these are not specifically stem cells. These are, because beyond stem cells, there are a lot of reporter mice that have GFP in almost any imaginable cell in the body. Because GFP, right, is a fluorescent protein and it allows you to track any cell that you're interested in. And in this image, this is actually from the GenSat project at the Rockefeller University. So when we saw these, we realized, oh my God, then maybe what we have to do is just to make a mouse whose T cells recognize GFP. And then GFP becomes a stem cell specific antigen. So this is what we did. So I took mice, I vaccinated these mice with GFP. I got this GFP specific CD8. So everything is in the context of CD8, cytotoxic T cells. I FACS sorted these GFP specific T cells. And since the TCR is already rearranged in these T cells, right? It has already happened, and there's no, right, like unrearranged gene in these cells. Back then there wasn't TCR single cell sequencing yet. What we decided to do was, rather than trying to clone the TCR, like old school, we had this idea, actually Brian had this idea, of performing somatic cell nuclear transfer. And the way this works is, as I said, the T cell that is specific for GFP, it already has the rearranged TCR. So what we did is, I literally took these T cells in the New York City subway. I went uptown to the laboratory of Dr. Judith Agudo at the University of Columbia and also New York Stem Cell Foundation. And he took the nucleus of one of these T cells, transferred this nucleus into an oocyte that had been enucleated, and basically generated stem cells from the T cell that then we injected in a blastocyst bag at Sinai, and to obtain a mouse. So we basically cloned a mouse from a GFP-specific T cell. And once this mouse was generated, this mouse was a just eGFP death-inducing T cell mouse. And therefore we call the mouse, as Matteo alluded to, as the Jedi Mouse. So once the mouse was generated and we were ready to go, first thing was testing whether these T cells indeed recognize GFP. The first model I used was the one I knew the best, which is beta cells. We knew that beta cells are susceptible to T cell killing. So if the mouse and the T cells work, we should obtain killing with beta cells that express GFP. And the Jackson Laboratories has a mouse that expresses GFP in the beta cells here. So I injected these Jedi T cells, and sure enough, the T cells go to the pancreas, they kill the islets, and the mice develop type 1 diabetes, they develop hyperglycemia. So this was actually neat because it showed to us that the tool was much more versatile than we had anticipated. We made the mouse thinking of, right, like this stem cell goal, but we had developed a mouse that could be used to model any autoimmune disease as long as a GFP mouse existed for the cell that one was interested in targeting. Something else that was very, rewarding or interesting to see was that when you have an antigen that you can see, first of all, you don't need to do any staining. So for example, this islet here, this is not stained, this is the native GFP, which means that you can also see live cells because you don't need to stain to see the antigen. And then another thing is that when you see the antigen, for example, here, this one beta cell, now it's very apparent. I know it has the antigen. It's surrounded by T cells, and yet it is alive. So how is this beta cell still alive? And actually, this is one of the things that I had observed as a postdoc, and I sort of like left aside. And we are recovering or regaining interest now in my lab. Something else is trafficking. Here, this is a mouse model that expresses GFP in microglia. We could see that when you have Jedi T cells, they are actually able to cross the blood-brain barrier and go to the brain and kill microglia. So it was, and actually I did this experiment before the lymphatics of the brain were published. So you can see T cells cross the BBB, and this tool can also help anyone study trafficking wherever one is interested. And lastly, we realized that actually these T cells could be used as what we like to call cell knockouts. So if you have a mouse model that has GFP in a cell type, and you want to study what the role of that cell type is, you can deplete the cell type. And there are models like the diphtheria toxin receptor transgenic mice to do this approach, but they don't exist for every single cell. So if you do have a model that has GFP in a cell that you are interested in, you can use T cells, which are nature's, I guess, DTR, and interrogate the role of that cell. This is just an example where I had FOXP3 EGFP mice, and when I injected Jedi T cells, the Tregs got depleted. And the mice developed, right, like all the features, splenomegaly, right, like all the inflammatory and autoimmune disease aspects, exactly the same. It phenocopied exactly the same as the FOXP3 diphtheria toxin receptor FOXP3 DTR mouse. So the Jedi T cells can be used as a depletion tool. And then going back to what was really my interest was, okay, what about stem cells? Now I do have the perfect tool to study how stem cells interact with T cells and whether or not they are immune privileged. And to do so, I took advantage of LGR5 GFP mice. These are the mice that were developed by Hans Clevers in the Netherlands that express GFP in various types of stem cells. So when he made the mouse, it was basically to study intestinal stem cells, which are here in the crypts. But later on, himself and other groups have found that LGR5 GFP mice also have GFP in other epithelial stem cells. So the first stem cell that I decided let's go and take a look were the intestinal stem cells. As I said, they are here in the crypts. And the role they play is they are highly, highly proliferative because they are constantly replenishing the epithelial layer, which is a tissue with a very fast turnover, right? Like every few days, the whole epithelial layer in your intestine is new. So the stem cells to me seemed like very important. So I thought, okay, if there is a stem cell that is immune privileged, I bet you this is the one. So how did I go about this? I took these LGR5 GFP mice. Here you can see the GFP cells. This is how they look like. I injected Jedi T cells. What happens? Well, they got killed super fast and super, super efficiently. So first of all, the big dogma in the stem cell field that stem cells are immune privileged, not true. At least there's this one very critical stem cell type in the intestine that is not immune privileged. And then it opened the door to, okay, like this one is not immune privileged. What about the rest, right? Like, let's see what happens. Okay, second type, these mice express GFP in stem cells in the epithelial layer that surrounds the ovaries here. These stem cells are, also important. They actually are also quite high proliferative because what they do is they replenish this epithelial layer that is broken every time an oocyte is released. And I thought, ovaries, these are, these very special places, right? They contain the oocytes. These ones are likely the ones that are going to be immune privileged. Are they? So this is how the fax plots from these ovaries look like. Here you can see the GFP cells. And when I injected this Jedi T cell, boom, again, everything got killed. Again, super fast, super efficiently, not even one left. Okay, so second stem cell type that is not immune privileged, killing again the dogma I was referring to. So what about the stem cells in the skin? So these mice, the LGR5 GFP mice, they have GFP in a population of stem cells in the skin called hair follicle stem cells. So these hair follicle stem cells, as the name says, they are in the hair follicles here. And they are quite, you know, like inactive, let's say. They are quiescent for a long time during, you know, because hairs have cycles. So when the hair is not growing, the cells are there not doing much. And then when the hair goes into cell cycle, then the cells proliferate and they help in the growth of these hairs. So what happens in this case? And I did the same thing. So in these mice, you have here the GFP cells, you inject the Jedi T cells. And to our big surprise, these hair follicle stem cells did not die. And I have to confess here that, you know, approaching this from a, you know, immunology perspective, I was like, what? Like intestinal, ovarian die, the hair follicle stem cells, they do not. And when I presented this in the work in progress at the Immunology Institute at Mount Sinai, you know, the reaction in the audience was pretty much the same, like, oh my God, the hair follicle stem cells. And this is all published, so I'm not gonna go into lots of details, but to make a super long story as short as I can, what we found is it's not Treg dependent, it's not lack of trafficking. The skin is not an immune privilege site, right? Like the skin is actually the opposite, if anything, right? Like there are lots of autoimmune diseases that happen in the skin. So what was happening? And how is, you know, this even possible? And as I said, these stem cells, they all express LGR5. They are all dependent on wind. They are all epithelial. What is different besides the site between the hair follicle and the other stem cells? And we realized that actually cell cycle was a big difference, that the ones in the skin were highly quiescent, whereas the other ones were highly proliferative. Could this actually be linked to the immune evasion that we were seeing? And this is just to illustrate what I was saying. So Hodge to show the cells that are undergoing S and G2 in cell cycle. And you can see these hair follicle stem cells are extremely quiescent. So could this be the case? How do you go about testing this? And what is neat about the model of hair follicle stem cells in the mouse is that all hair follicles are synchronized, which means that when the hair grows, it's the same in all hair follicles. So all the cells are proliferating. And when it's in the resting phase, they are all in the resting phase. So what I had to do was to find a moment when these hair follicle stem cells were growing and test whether they then had lost the immune evasion phenotype. And this is exactly what I did. So when the mice are younger, the hair follicles are growing, and it's called the anagen phase. And when I injected Jedi T cells into these mice in the anagen phase, then you can see that now for the first time, these hair follicle stem cells got killed. And what was the mechanism? How were these ones killed, but not the other ones? So we did what I feel like, you know, many of us do when we don't know where to find the mechanism, is we performed RNA sequencing. And what we saw was that this telogen, this is the resting phase. So when hair follicle stem cells were quiescent, they had a massive downregulation of MHC class 1 and the entire machinery of the antigen presentation. And when these hair follicle stem cells entered into growth, they started a proliferation program, then everything got back to being expressed. So just to summarize, what this final work in my postdoc showed was that stem cells are not universally immune privileged. However, some of them are. I showed you about hair follicle stem cells. I didn't talk about them, but they are also in the published work. Satellite cells in the muscle, they are highly quiescent and they are also immune privileged. And conversely, as I was mentioning, the proliferative stem cells are susceptible to T cell killing. So once this work was published, I was left with, you know, this feeling that these, right, like mattered beyond the field of stem cells, that this was also important for cancer biology. And I had specifically these two questions. Is this something these processes of immune evasion that cancer stem cells hijack, because cancer cells, that's what they do. They co-opt mechanisms that are already in place. And are quiescent cancer cells actually immune evasive? There is this phenomenon that it's well known that, right, like patients can have a tumor, the tumor is resected and eliminated, and things seem to go well, but then years later, there is a relapse with, you know, the same cancer regrowing in the lung or in the liver or somewhere else, which indicates that some tumor cells were able to hide and stay dormant or quiescent for a long time. So the fact that these cells survive for this long time means that the immune system was not able to kill them. Was this something that could, you know, be parallel to the findings I had during my postdoc? And with these ideas, then my trip was a little shorter, actually by car. Well, you know, we got all our stuff, we left New York, and we moved to Boston to start the lab at the Harvard Medical School and then at Harvard. And then here, my lab, as Matteo was saying, we study immune evasion of stem cells. We are still trying to engineer stem cell products such as stem cell islets for immune evasion of the immune system for treatment of type 1 diabetes. So everything we learn now we can leverage to protect these cells. And we are also interested in how tissue stem cells, when they acquire oncogenic mutations, how they interact with the immune system, right? So in the skin, as I was talking about, in the intestine as well, right? These LGR5 cells are not immune evasive. And we know they are the cell of origin of colorectal cancer. How do they become immune evasive? And lastly, metastatic stem cells, they share lots of features with tissue stem cells. So how are they also acquiring these immune evasion phenotypes? And now I'm gonna present some of the most recent work in the lab. This was done by a super talented, brilliant, and driven student, Pilar Valdominos. She's also from Spain. She came to the lab because she saw me present my work once, and she thought the Jedi Mouse was very, you know, a very interesting technology, and she wanted to use it to study immune responses in breast cancer. So what she did is she developed cell lines, triple negative breast cancer mouse lines that express GFP, with the idea of injecting these mice with Jedi T cells and study immune evasion in this context. And this is, you know, the first experiment she did. As I said, she injected these tumor cells with GFP and Jedi T cells. And, you know, I remember the first experiment she did, she came to my office, you know, like, Judith, I think your T cells are not working anymore because the tumors shrunk a little bit, but then the ones that had been treated with Jedi, the tumors in these mice, they shrunk a little bit, but then they came back again. But of course, this is cancer, right? It's not like healthy cells. So T cells have a tougher time trying to kill them. And then I said, okay, let's take a look. Let's open and see what is happening. And what she saw was that in the untreated tumors, in the untreated mice, the tumors were, you know, all GFP positive, all cells were positive. However, in the tumors that came from mice that had been treated with Jedi T cells that kept growing, there were cells that were GFP, but also lots of cells that were GFP negative. And this is not something, you know, new that we had discovered. This is immune editing, right? Like when there is a very strong immune pressure, cells that, for example, don't have the antigen or that lost the antigen, they have a growth advantage, so they keep growing. But this gave us an idea, which is that when we study immune, you know, resistance to immunotherapy, and we are not able to visualize the antigen, we are mixing lots of things without realizing, right? Like, let's say she didn't have GFP and we were giving checkpoint blockade, and this is actually what was happening, but we didn't know, right? We would be mixing cells that were escaping through different mechanisms, because this tumor cell here is surviving because it no longer has GFP, so the T cells cannot see it. Whereas this tumor cell here has GFP. it should get killed, and yet it's not killed. So this one has evolved some other mechanism of resistance. And now by virtue of being able to use this GFP, we can separate these two completely different biological processes of immune evasion and interrogate these cells separately. So this is what Pilar did, and to have it, in a much easier way to do it, what she did is she used, again, these GFP breast cancer cells and mixed them with the same cell line, but instead of GFP with mCherry reporter, which is completely different from GFP. The sequence is different, and it's not recognized. So this was to label non-targeted cell. They were mixed together, and now she could FACS sort these GFP positive cells that we know should get killed but are not, which means that they have evolved immune evasion mechanisms other than losing the antigen. And she could compare with cells within the same tumor in the same microenvironment. So these cells are exposed to the same cytokines, same inflammatory milieu, and it's the bulk of the cells. To see to interrogate and to identify the mechanisms of evasion from these cells. And this is how it looks like. So it doesn't matter how you do it, you always have a pocket of GFP cells that survive. How are these guys not getting killed? The first thing, obviously, was to look at MHC class 1, because they do have the antigen, but maybe they lost the ability of presenting it. And actually, that's not the case. MHC was retained. So they should get killed, as I was mentioning. What is the mechanism of immune evasion? So we looked at all the possible suspects. We also used PD-1 knockout Jedi T cells to circumvent PD-L1 as a mechanism of suppression. What could be happening here? And again, we sequenced this. We took advantage of a fluorescent antigen that allows you to isolate live cells and perform bulk RNA sequencing. And what we saw was, we were looking for some sort of, amazing immune-related gene that, was, there with your, telling you, this is the immune evasion mechanism. This is not what happened at all. And when we performed gene ontology analysis to have an unbiased approach to, better understanding what was happening, it was surprising to see that every single pathway at the top of the most significant p-value had to do with cell cycle. So if there was anything to learn from these surviving GFP cells, was that they seem to be either very slow growing or quiescent. And these are some of the genes. So you can see, for example, Ki67 is very low and almost any other gene in the pathway. But one thing is transcriptome, another thing is function. So are these cells really quiescent? And here what Pilar did is she took, again, mice, injected them with this mix of cherry and GFP cells. And these tumors grow pretty fast. So it's usually around 2 weeks. And it's an exponential growth. So at the beginning, they grow slowly, and then they grow very fast. What she did is during the last week of tumor growth, she injected EDU, which is a nucleotide analog of thymidine, to label cells as they enter into the S phase, into DNA replication. And she gave EDU every day for 1 week, which is, as I said, it's the last week, and it's the week of exponential growth of these tumors. So this is actually a long time for these tumors. As you would expect, the cherry cells had lots of labeling because, again, it's 1 solid week. Lots of cells were EDU positive. But then what was shocking and very rewarding to see was that the GFP cells that survived did not get labeled, which means that it's not only that they were not slow growing, they were actually literally quiescent for a week. So these cells did not grow for a week, which was very interesting to see. So there are these pockets of clusters, because they were aggregated of quiescent cells that did not grow. Now the problem was, okay, how do we figure out the mechanisms of immune evasion they use? Because we know class 1 is there, the antigen is there. How are they doing it? And the problem is EDU was not a very good tool for us to use, because EDU requires fixation of the cells. And we wanted to have a fluorescent reporter so we could manipulate, sort, profile live cells. We tried many things till we found this paper where they had developed this reporter, which is a fluorescent protein called mVenus that is fused to a truncated form of P27. So P27 is degraded as the cells go into cell cycle, and it's not degraded when the cells are in G0. And since mVenus is fused to this P27 is not functional, but it gets regulated in the same manner as the endogenous P27, which means that this mVenus is not degraded and is highly, it's very bright during G0. So it's a good tool to label quiescent cells. We obtained this construct and engineered, Pilar did, she engineered the breast cancer cell lines. And the first thing was to confirm whether the construct behaves in the way, right, that should in our context of breast cancer. And this is a primary breast tumor. And you can see the mVenus P27 reporter and Ki67, they very nicely exclude each other. So it looks like it was working. But then the question was, okay, we know that this P27, for example, here is labeling a cell that right now in this very moment is in G0. But was this cell in G0 1 hour ago, 3 hours ago, 3 days ago, right? Like, how do we know? Are we actually seeing the same cells that we saw after Jedi killing? So what Pilar did is she took this cell line that has the P27 reporter without GFP, without any T cell pressure. She grew these tumors again, and she treated the mice with EVU, with this thymidine analog, for 1 week. And as you can see, is that lots of cells got labeled after 1 week, as you would expect, but the P27 Venus cells did not get labeled. So for 1 solid week at the end of tumor growth, these cells had remained quiescent, indicating that we are talking about the same cells. So now we have the perfect tool to study how they are surviving T cell killing. And I'm not gonna show it, but something that she also did, Pilar did, was to directly have the reporter with GFP and trying to kill these cells with Jedi T cells in the primary tumor. And they survived. They are the ones that survived the best, these P27 positive cells. So how is this survival happening? Once Pilar clearly demonstrated that they are surviving, right, even in a clear antigen specific manner with Jedi and GFP, we decided to take a step back and see, okay, maybe it's a problem of infiltration, because we could see that the GFP cells and the P27 form clusters. And this is how a triple negative mouse tumor looks like if you stain for CD3 to see T cells. T cells, and this is well known, T cells are not homogeneously distributed in a tumor, right? Like the infiltration is heterogeneous, right? Like some areas have more T cells, some areas have very few T cells. What was really, really, really interesting and again, shocking for us was that when Pilar overlaid here the P27 reporter, I hope you can appreciate that actually it nicely correlates with the areas of low T cell infiltration. So it looks like P27, these quiescent cancer cells are a major, I'm not saying but we do think it's a major driver of T cell exclusion, as you can see here. And obviously this is all mouse, and you might think, okay, what about patients? Something great of being in a place like the Dana-Farber Cancer Institute is the close contact with physicians and actually how interested they are in basic research and how supportive and the crosstalk and the communication between basic researchers and oncologists. So we obtained samples from our colleagues here in the breast cancer program, and human tumors look very different from mouse tumors, but still Pilar could see that in regions with lots of P27 positive cells, there were no T cells to be found, as opposed to regions with no P27 tumor cells where T cells were clearly infiltrating. And this was true within different regions from the same patient, but also from patient to patient. So it looked like there was indeed a correlation between proliferation and infiltration, or rather lack of proliferation and quiescence. Sorry. So this seems to be true, which like made us wanting to learn more. But something that was not criticized, but when Pilar presented her work at the Work in Progress here to an audience of immunologists, a question was, oh, but if the cells are quiescent, are they senescent? Are they terminally, sorry, there's a helicopter. Are they terminally, arrested in cell cycle? And if so, is it really a big deal that the immune system is not killing them, right? Because again, as an immunologist, if a cell is there sitting in a quiescent state and not doing anything, maybe it's okay if the immune system fails to kill them. To test whether or not these cells are, as I said, arrested and whether or not they are able to regrow tumors, what Pilar did is she took advantage of the fact that we had a visible reporter, so cells can be sorted live. And she grew tumors that had M-Cherry as an ubiquitous reporter, and then M-Venus P27 to see the quiescent cells. She took these tumors, digested them, and sorted the ones that were P27 negative, so proliferative cells, and the ones that had P27, so the quiescent cells. And she re-injected these sorted cells into mice, and she saw that they all regrew tumors, which made us wonder, okay, what if we actually inject fewer and fewer? What are we gonna see? And to our surprise, not only did these quiescent cells regrow tumors, they actually did it more efficiently than the proliferative counterparts. So this experiment was done injecting 500 cells of either cell line. So this was actually, and was true across different cell lines of breast cancer, mouse cell lines. And as you can see, with 500 cells, the quiescent cells regrew into tumors, whereas proliferative cells from the same tumor did not. This means that these cells are actually dangerous and capable of regrowing tumors. So we better learn how they are escaping from immune recognition, because we for sure need to get rid of them. Plus, quiescent cells, this is known, are resistant to chemotherapy, because chemotherapies are basically poisons that kill proliferative cells. So we needed to understand what was happening here. And again, she sorted the cells. We performed RNA sequencing. We saw some signatures that had to do with chemoresistance, pointing out again to the fact that probably these are the same cells that escape during chemotherapy. And we saw some genes related to stemness, which could explain the improved tumorigenic potential. Something else that we saw in these cells was a clear signature of hypoxia. So it seems like HIV-1 alpha probably was a major driver of this phenotype. And this was great, but it was not telling us how they were escaping. So again, we decided to go back to the image I was showing before, where we see that these P27 cells are excluding T cells. And then here we had, actually, we realized that the question was in reality two. One is, how are these quiescent cells keeping these T cells away? This is one question. And then the second question is, it's not that all T cells are always away. A few of them do enter, and as you can see here, they are in close proximity to the quiescent cells. So how come this T cell is failing to kill this quiescent cell? This is the other question. And something that has become, streamlined, used by everybody to figure out, mechanisms and to understand heterogeneous tissues and complex biology is single cell sequencing. The problem is, if we take this tumor, digest it, and sequence, we are not able to learn anything, because we need to know where the cells came from. If we want to profile, for example, the T cells in this tumor to understand the connection and the crosstalk with these quiescent cells, we need to know that this T cell came from a region of proliferative tumor cells, as opposed to this T cell that came from a region of P27 cells. How do you go about that? And this actually, what happened here was that we were inspired by a technique and a very beautiful paper that Matteo published in Science, where they described a technology called niche-seq. And with the help of Dr. Olga, oh, you cannot see her here. Olga Barreiro, she's a senior postdoc in Uli von Andrian's lab. Super smart, super talented. If any institute is looking for a great immunologist as a new faculty, she's your person. So with the help of Dr. Olga Barreiro and inspired by this great work from Matteo's lab, we decided to do something similar, but tweak it so we could use it for our model. And we used, we call this technique photoconversion of areas to dissect microenvironments. So Padme-seq, to follow up with the Star Wars saga. And the way this works is the following. We had mice that express the photoconvertible protein caide. This protein is green, and when you shoot UV light, the protein goes to red. So cells go from green to yellow because the conversion is never in 100% of the proteins. So you have a mix of green and red, so the cells become yellow. And then Pilar, she re-engineered her cell lines to have a reporter that in this case, instead of Venus, which is yellow, was tomato, so it's red. And these tumors have no color, no ubiquitous marker, just the tomato P27 reporter. So these clusters of quiescent cancer cells were red. She grew these tumors, and then what she brought new to the table was that we had this idea of using a compressed tome, which is like a microtome or a cryostat, but different, that you can get thicker sections of live tissue. And it's broadly used by neuroscientists to obtain sections of brain. In our case, we used it for tumors. So Pilar embedded these tumors in agarose and got these 200 or 100 micrometer thick slices of tumor. And now under the microscope, she could very nicely see these clusters of quiescent cancer cells, and she could label everything that was inside these clusters of quiescent cancer cells in one section. And in the section consecutive to this one, so from the same tumor in an adjacent region, she labeled the cells in the regions of proliferative tumor cells. So we could compare the infiltrates and the stroma of these two components within the same tumor. And then once the cells were labeled, she digested these slices and fax sorted the cells and submitted for single cell sequencing. Oh, why isn't it? Oh, here. And this is how it looks like. So this is one of these clusters of quiescent cancer cells in red. And these are the caide cells from the host mouse that infiltrate the tumor. So they are in green. What is beautiful about photoconversion is that you can draw any shape you want under the microscope. And then these cells, as I was alluding to before, they become yellow. So now when you digest the tissue, you can fax sort these cells. And now you know that this macrophage or this fibroblast here that we are fax sorting comes from these regions and these clusters of quiescent cancer cells as opposed to other fibroblasts out there. So we can then compare and see what's different that allows for this T cell exclusion and lack of killing. So this is the UMAP that we obtained after, with all the data from inside and outside these clusters of quiescent cancer cells. And we could see that we could see all the well-known subsets that suspects that infiltrate these tumors in mouse models and even in patients. And the first question was, okay, what's different about fibroblasts? Because since we were seeing exclusion of T cells, we thought that probably fibroblasts were one of the populations to blame for that. And we did lots of analysis, but I think this single heat map summarizes our findings very well. So these genes are a signature that was published by Dr. Shannon Turley at Genentech, who is a worldwide renowned expert in fibroblasts, in tumor, and the role they play in tumor immunology. So anyway, they found this signature in suppressive fibroblasts that can predict lack of response to immunotherapy. And this is usually the marker they use, LRRC15. And what we see is that these fibroblasts, they are actually sitting or living or happening next to these quiescent cancer cells. So now we know where they are within the tumor. Whether they are the ones inducing the quiescent phenotype or the quiescent cells are the ones inducing this phenotype, we don't know yet. What else? Well, we wanted to look at T cells because the fibroblast phenotype could explain the exclusion, but what about the T cells that do make it to the clusters and don't kill? What's wrong about them? We took a deeper look into CD8 T cells, and we identified also, well-known and well-described subpopulations of CD8 T cells, from superterminally exhausted to progenitor exhausted, and proliferative T cells. What is happening here? And what we found was that here, the terminally exhausted T cells, which are the ones that are the most dysfunctional and incapable of killing tumor cells, are highly enriched inside the quiescent cancer cell clusters. So these cancer cells are surrounded by completely dysfunctional CD8 T cells. And then conversely, proliferative CD8 T cells, which are the ones that could do the job, were almost not found at all close to these quiescent cells, showing this dichotomy. And what was very interesting as well is that we know that these terminally exhausted cells exist. Lots of labs are working on these populations, trying to understand, how they arise and, the epigenome and all of this biology. But at least to the best of my knowledge, no one has described where within the tumor they localize. There are some works now, for example, Thorsten Mempel published a very beautiful paper showing that CXCL16 can recruit the least dysfunctional cells. But in any case, here we were able to identify that these TIM3 positive, PD1 positive, terminally exhausted T cells are located close to these quiescent cancer cells. What else did we find? Okay, how are these, the terminally exhausted T cells happening in these regions? We had seen that the quiescent cancer cells had a very clear hypoxia signature. So then we decided to take sort of an unbiased approach and ask what populations had also a very clear hypoxic score, thinking that maybe they could be drivers connecting the quiescent cancer cell phenotype to the dysfunctional niche for immune cells. And as you can see here, actually the top cells were plasmacytoid dendritic cells. So dendritic cells. So plasmacytoid dendritic cells were the top. Cross-presenting DC1s was the second, and then this population that was described by Miriam Merat, others have called them other ways, MREG DCs were the third. So clearly dendritic cells seem to be profoundly affected by the hypoxic environment that these quiescent cancer cells were generating. And I have this slide mostly for non-immunologists to illustrate how important dendritic cells are. Because in the world of cancer, everyone knows about T cells and there's a big focus on T cells. And we often forget about this tiny and yet extremely powerful population of dendritic cells. And the way I explain this to patient advocates here that I interact with is that T cells are the soldiers, right? With the guns that go there and kill the bad guys. But dendritic cells are the general and, like the big bosses in the army that instruct the T cells and educate them and tell them what to do. Dendritic cells actually are so powerful that they not only can activate and induce a T cell response, they can also induce energy and tolerance, which is actually what I had been doing at the very beginning of my postdoc. So they're very, very important. And I love this tiny figure from this old paper from Ken Murphy's lab, because I think it illustrates really well the importance of these cells. So this is a knockout mouse for BATF3, and this mouse has a normal immune system, but is missing cross-presenting dendritic cells, which are the dendritic cells that activate CD8 T cells. And they have a model where they grow tumors that are super responsive to immunotherapy. And then when you give this tumor to a wild-type mouse and you give immunotherapy, the tumor regresses and the mice get cured. If you grow this tumor in immune-deficient mice that don't have T cells, such as rat knockout mice, and you give immunotherapy, since there are no T cells, the tumor keeps growing. And if you do the same in mice that don't have cross-presenting dendritic cells, but do have T cells, and then you give immunotherapy, boom, the tumor keeps growing as well. So clearly these little guys are extremely important. What is that we found in our dendritic cells? So what we found was really amazing. We found that across the board, all dendritic cell populations showed a very obvious signature of dysfunction and signatures that linked to the lack of an immune response. So for example, these MREC disease, which are very important because these are the ones that take up tumor antigen, they had a downregulation of IL-12, which is known to be essential for triggering a T cell response, downregulation of the antigen presentation genes, which means that they cannot present well antigen to CD8 T cells. FASN1 is a gene that most likely indicates an impairment in migration. So dendritic cells, they need to migrate to the draining lymph node to educate T cells. Plasmacytoid dendritic cells also showed an abnormal signature with key transcription factors like E22 downregulated. Same thing for all of them. And this is just an image because I don't know how you say this in English. We say an image is worth 1,000 words. At least that's the literal translation from Spanish. I think it makes sense in English as well. Anyway, so in an image, I think I hope I can convince you that, so this is the cluster of quiescent cancer cells in yellow. And we are using CD11C here as a surrogate for dendritic cells. It's not a good marker by itself. We were actually using F480 to gate out macrophages. So because macrophages in tumors can have CD11C. Anyway, so CD11C to visualize dendritic cells, and then class 2 as a surrogate for functional and properly functional dendritic cells. And as you can see, CD11C positive cells that were outside these niches of quiescent cancer cells, they had lots of class 2, whereas this CD11C here, inside these clusters and in close contact to these quiescent cancer cells, they had much lower class 2, further proving this phenotype of dysfunctional dendritic cells when they were in close contact to these quiescent cancer cells. So as a summary, what Pilar found was that in breast cancer, there is this population of quiescent cancer cells that forms niches where T cells are reduced in number. The few T cells, for example, this little guy here, they are extremely exhausted. They are TIM3 positive, highly dysfunctional. There are also dysfunctional dendritic cells, which we think is what drives this dysfunction of the T cells. There are highly suppressive CAFs that most likely explains the clear immune exclusion. And all of these leads to survival of these quiescent cells that, since they are capable of regrowing tumors and actually very efficiently, we think that they represent reservoirs of resistance to immunotherapy. And finally, I just want to thank the members of my lab, obviously Pilar, who was, an unstoppable force of nature driving the project, other students in my lab, recently recruited postdocs, collaborators, computational collaborators in the breast cancer program, collaborators that work with us with stem cells, Yachi Shu for the skin, Omer Yilmaz in the intestine. We started work with Douglas Melton for stem cell derived islets, but now he's gone to industry. So we are a bit orphaned there. My mentors, female faculty around that have been an amazing support for us and funding sources. And I'm looking for postdocs. So if anyone is interested, I try to have a lab that, everyone feels welcomed. Sorry, did I talk for too long? I did.
No, it was wonderful, Judith. Hi, how are you? I joined a little bit late to your talk, but I was able to enjoy how wonderful work you shared with us today. Thank you so much. So many amazing insights for the stem cell biology and then for cancer immune evasion, and terrific, really. Thank you so much, and so engaging too. So yeah, really appreciate that you have given the Global Immunotalk today.
No, thank you for inviting me. Really an honor.
And I'm sure you will have emails from postdocs. It would be a privilege to work in your lab. So that's sure we'll follow up. Okay, so just as you know, questions are via Twitter. So if you would like to interact with Judith in Twitter, please search for the account Global Immunotalks, find the tweet that says ask questions for Judith Agudo here, and then you reply to that tweet with your question and mention the hashtag Global Immuno, and Judith will use her own Twitter account to answer the questions. And so you can ask the questions right now before the talk, or you can ask after if you watch the talk in YouTube. And like always, thank you everyone for joining. Hope you join again next week. And thank you, Judith, again for a beautiful talk.
Thank you. Thank you so much.
Take care.